Hello everyone, my name is Harika, studying third year of biotechnology. Today I am doing a video about gene libraries. Gene library, construction, cDNA library and genomic library, screening of gene libraries, screening by DNA hybridization, immunological assay and protein activity. Marker genes, selectable markers and screenable markers, non-antibiotic markers. Gene expression in prokaryotes. Tissue-specific promoter, own inducible promoters, strong and regulatable promoters, increasing protein production, fusion proteins. Translation expression vectors. DNA integration into bacterial genome, increasing secretions, metabolic load. Recombinant protein production in yeast. Saccharomyces cerevisiae expression systems. Mammalian cell expression vectors, selectable markers. In gene libraries, there are two types, cDNA library versus genomic library. These are the representation genes of genes in expression, genes in expression, and total gene. Now, genomic library compared to cDNA library. Genomic library. A gene library is a set of clonal fragments that collectively represent the genes of particular organism. Particular genes can be isolated from DNA libraries. Both genomic and cDNA libraries can be screened by hybridization. cDNA libraries. Complementary DNA libraries are generated by the reverse transcription of mRNA. cDNA is representative of the mRNA population and therefore reflects mRNA levels and the diversity of splice isoforms in particular tissues. The PCR can be used as an alternative to cDNA cloning. Full-length cDNA cloning is facilitated by the rapid amplification of cDNA ends. cDNA libraries is complementary DNA prepared by reverse transcribing cellular RNA. Cloned eukaryotic cDNAs are useful because they lack introns and other non-coding sequence present in the corresponding genomic DNA. Introns are rare in bacteria but occur in most genes of higher eukaryotes and are transcribed by RNA polymerase as primary transcripts that goes through a series of processing events in a nucleus before appearing in the cytoplasm as mature mRNA in a process called splicing. Introns may occur in the 5' or 3' untranslated regions of intron sequence by a process called size of DNA clone is significantly small than that of corresponding genomic clone. For example, the human, and here are the example of size of cDNA. Construction of gene libraries. A DNA library or gene library is a collection of DNA clones gathered together as a source of DNA for sequencing, gene discovery, or gene function studies. Depending on DNA source, library can be grouped as genomic library or cDNA library. Genomic library are produced when the complete genome of the particular organism is cleaved into thousands of fragments and all the fragments are cloned by insertion into a cloning vector. The first step in preparing a genomic library is partial digestion of the DNA by restriction endonuclease, such that any given sequence will appear in fragments of a range of sizes that are compatible with the cloning vector and ensures that virtually all sequences are represented among the clones in the library. Fragments that are too long or too small for cloning are removed by centrifugation or electrophorese. The ligated DNA mixture is then used to transform bacterial or yeast cells to produce a library of cell types, each type harboring a different recombinant DNA molecule. Ideally, all the DNA in the genome under study will be represented in the library. Steps for construction of cDNA library. The synthesis of double-stranded cDNA suitable for insertion into a cloning vector involves three major steps. First one, First-strand DNA synthesis on the mRNA template by reverse transcriptase. Removal of the RNA template. Second-strand DNA synthesis using the first DNA strand as a template by DNA-dependent DNA polymerase 1. All DNA polymerases, whether they use RNA or DNA as a template, require a premier to initiate strand synthesis. Of several alternative methods, the one that became the most popular was that of Maniatis in 1976. This involved the use of oligo-DT premier annealing at the polyadenylate tail of the mRNA to prime first-strand cDNA synthesis and took advantage of the fact that the first cDNA strand has the tendency or transiently fold back on itself, forming a hairpin loop, resulting in self-priming of the second strand. It is the representation of nucleus cloning vector. Improved method for cDNA cloning. A serious disadvantage of the hairpin method is that cleavage with S1 nucleus results in the loss of a certain amount of sequence at the 5' end of the clone. This strategy has therefore been superseded by other methods in which the second strand is primed in a separate reaction. The first strand is tailed with oligo DC, allowing the second strand to be initiated using an oligo DG premier. Screening gene libraries. There are two types, genomic and cDNA. By nucleic acid hybridization, most commonly used method of library screening because it is rapid. It can be applied to very large numbers of clones and in the case of cDNA libraries can be used to identify clones that are not full length. Screening by PCR is possible if there is sufficient information about the sequence to make suitable primers. Screening expression libraries. If a DNA library is established using expression vector, each individual cloning can be expressed to yield a polypeptide. Huh. Nucleic acid hybridization was first developed by Grunstein and Hognes in 1957 and modified by Hanahan and Messelson 1980. The colonies to be screened are first replica plated onto nitrocellulose filter disc that has been placed on the surface of an agar plate premier to inoculation. A reference set of these colonies on a master plate is retained. The filter bearing the colonies is removed and treated with alkali so that the bacteria colonies are lysed and the DNA they contain is denatured. The filter is then treated with proteinase K to remove protein and leave denatured DNA bound to the nitrocellulose, for which it has a high affinity in the form of a DNA print of the colonies. The DNA is fixed firmly by banging the filter at 80 degrees C. The defining labeled RNA is hybridized to this DNA, and the result of this hybridization is monitored by autoradiography. A colony whose DNA print gives a positive autoradiography result can then be picked from the reference plate. It is a representation of detection of recombinant colonies by colony hybridization. Please take a look. Screening by gene libraries by PCR. Library screening by PCR is demonstrated by Takumi and Loris in 1994. Instead of plating the library out on agar as would be necessary for screening by hybridization, pools of clones are maintained in multi-well plates. Each well is screened by PCR and positive wells are identified. At last, these are the applications of gene libraries used. First one, used to collect and store information as a set DNA molecule. The cDNA library are used for isolating gene that codes for a particular mRNA. It is used to isolate DNA sequence to code mRNA. Genomic DNA sequence is used for microarray analysis. Thank you guys for watching.